It's great honor for me to be here as a recipient of Nobel Prize in Physiology or Medicine. First, I would like to express my sincere appreciation to Karolinska Institute and Nobel Foundation for this wonderful opportunity. And I would like to say thanks to all the audience here today. Let's begin. I believe science is a system of knowledge that is gradually accumulated by society, but it's also inherently human activity. I believe that every scientist is a product of the era which live. So today I would like to start my talk from a brief introduction of my life, then give us a historical overview of our work. I was born in Fukuoka, southern Japan in 1945, half years before the end of World War II. This was a very challenging time in Japan, and everyone had difficulty getting basic daily necessities, including food. I myself suffered severe malnutrition and was a very sicky child. Around this time, my mother got tuberculosis and spent a long time period bedbound. But she was miraculously able to recover thanks to the gift of just developed antibiotics from a family friend in Hawaii. I remembered the word streptomycin and oleomycin without knowing what did that mean. My childhood home was surrounded by nature with rice paddies, streams, hills, and sea all nearby. I spent a lot of time outside catching fish and picking plants. In elementary school, I was engrossed in collecting insects and watching night skies. I would say this intimacy for nature had a strong influence on me. At that time, I vaguely longed to be a scientist. In high school, I joined to the chemistry club where I experienced the wonder of chemical reactions. I entered the University of Tokyo, where I initially hoped to be a chemist. But soon I had difficulty to find a field to work in. Thankfully, this was a time when central dogma was just being established. Molecular biology immediately fascinated me, so I decided to join the lab of Kazutomo Imahori as a graduate student. There, I began my research career studying ribosome, which is a machinery of protein synthesis. This period made me keenly aware of the continuous nature of protein synthesis within cell. I gradually became interested in uncharted function of biomembrane. So I worked on the mechanism of action of collagen E3, which is a cytotoxic protein that inhibits protein synthesis by binding a receptor on the membrane of other cell. After finishing my doctoral studies, I enrolled in the lab of Gerald Edelman at Rockefeller University, New York. There, during my final year in Dr. Edelman's lab, I worked with Mike Jarzynski on the initiation mechanism of DNA replication in yeast, which was my first encounter with yeast as an experimental organism. At the end of 1977, I returned to Japan as an assistant professor in Yasuhiro Andaku's lab at the University of Tokyo. Dr. Andaku's entire lab was tasked with studying transporters and respiratory chain in E. coli. But fortunately, he allowed me to start his work. After a period of reflection, I decided to work on the yeast vacuole. I'm not a competitive person, but rather prefer to work on a subject that is not factionable. This EM picture shows the yeast vacuole. You don't see any structure inside the vacuole. At that time, vacuole was thought just it's normal than the garbage dump in the cell, but I thought it must play yet unknown role in cell physiology. And I was able to show active transports of amino acid and calcium ion through the vacuole membrane, providing evidence that vacuole is important for homeostasis of metabolites and the ions. We also described the novel proton pump on the vacuole membrane, V-type ATPase, which generate a proton gradient across the membrane, vacuole membrane. In 1988, I was able to start my own lab, consisting just myself at the College of Arts and Science at the University of Tokyo. I decided to devote myself to lytic function of yeast vacuole. Since it is an acidic compartment and contains various hydrolytic enzymes, so I thought it might be homologous to lysosome. However, I had no concrete idea how to approach this problem. Before going to experimental details, I will briefly touch on protein metabolism in cell. Proteins, which are the polymer of amino acid, are the key player in all biological processes. Central dogma stated that the protein is synthesized according to the genetic information encoded in DNA through RNA. Since then, many researchers have studied hard to understand gene expression and protein synthesis. Recent progress in cell biology has revealed how each protein localizes to its specific destination in the cell. We know now each protein is ingeniously organized through the process of intracellular trafficking. However, one aspect that is missing from this picture was the degradation of protein within cell. When I held a biology class for first year undergraduate student, I used to start my lecture with this question: how many blood cells are made within just 1 second in our body? Calculation is simple, and given an answer about 3 million cells per second. How about hemoglobin within the red blood cell? Further calculation showed that about 1 times 10 to the 15, 1 million billion hemoglobin molecules synthesized per second. It followed that exactly the same amount of cells and protein are degraded. The key message is that living cells are so dynamically maintained. Japanese climate is characterized by 4 distinct seasons that influence our culture, and students at school learn that everything are continuously changing. Plant leaves offer an excellent example. In spring, leaves develop and then actively synthesize starch by sunlight. But in autumn, leaves turn yellow and fall off. What we see as a beautiful autumn color is actually caused by the degradation of leaves' photosynthetic machinery. Green chloroplasts are completely degraded and resulting amino acid transport to the trunk. Similarly, rice leaves turn yellow at harvest time. All protein in the leaves are degraded and transported to make proteins in rice grain for next generation. These examples demonstrate that degradation is not adverse process, but rather it's essential for new construction or regeneration. Let's think about the human body. Our body makes about 200 to 300 grams of protein every day, but we only intake about 70 to 80 grams of protein from the diet. The amino acid required for protein synthesis mostly come from the degradation of our body's own proteins. Life is maintained tightly controlled balance between synthesis and degradation, and recycling the essential for life. Early pioneer in the field of protein metabolism was Rudolf Schleiermacher, who in 1930s proposed the concept of protein turnover. However, unfortunately, his idea was not widely accepted. The monumental breakthrough come in the 1950s with the discovery of ribosome by Christian Dudu. Soon after, researchers at Rockefeller University analyzed the process of the delivery of our own cellular component to lysosome by electron microscopy, a process which Christian Dudu coined autophagy in 1962 from the Greek for self-eating. Glenn Mortimer and Parseglen afterward produced informative works by tackling both physiological meaning and regulation of autophagy in mammals. Despite this, our understanding of the molecular mechanism of autophagy remained in its infancy for many years. In the meantime, another intracellular protein degradation system, ubiquitin proteasome system, was discovered, as it became clear that this system plays important roles in the regulation of a variety of cellular processes. Protein degradation suddenly attracted a huge amount of attention in biological research. This was ultimately recognized by the Nobel Prize in Chemistry in 2004, which was awarded to these three scientists shown here. But the field of lysosome degradation and autophagy still remained quiet. Now let's return to my work. Assuming that the vacuole functioned as a Ricci compartment, the question I had were when, what, and how such plasmic constituents go across the vacuole membrane and become accessible to vacuolar enzymes. First, I searched what condition where massive protein degradation might occur in each cell life cycle. Spore formation is a meiotic process that generates 4 spores and is induced by the depletion of nitrogen, the essential element for amino acid. I reasoned that this dramatic cellular remodeling should require a degradation of pre-existing protein to make necessary proteins. I have to admit that I have always loved to observe each cell by light microscopy. The vacuole is relatively large, and it's only clearly visible organella in the cell. And inside of the vacuole is just a solid solution. So I knew that it is easy to see structure if they were inside the vacuole. I probably spent more time sitting at the microscope than anyone else at that time. I carefully observed the early stage of sporulation, but I couldn't see any significant change in the cell. I conceived to use a vacuolar proteinase deficient mutant to prevent further degradation. Fortunately, Elizabeth Jones had donated many proteinase mutant to East Genetic Stock Center. So I obtained this strain and shifted to the nitrogen depleted medium. Okay. Looking down the microscope, I saw many spherical structures vigorously moving around the vacuole. These structures appeared within 30 minutes and almost completely filled the vacuole after 3 to 4 hours. For me, this observation marked the exact starting point for more than 27 years of autophagy research. The reason I could discover this phenomena with only a common low magnification light microscopy or to the vigorous movement of this structure by Brownian motion in otherwise static yeast cells. This dramatic change was enough to convince me that I encountered an unknown and fascinating phenomena. Soon, Kazuhiko Takeshige showed this phenomena is not restricted to sporulation, but it's a general cellular response to a range of nutrient starvation condition. Then I called excellent electron microscopist Misuzu Baba and Masako Osumi to capture the morphology of this phenomena within cell by electron microscopy. Their effort produced beautiful and conclusive image of the entire autophagy process, as shown in the following several slides. This section image of nitrogen-starved cell at 3 hours, you can see spherical structure within vacuole. And this high magnification image shows these structures are bounded by a single membrane, and their contents are exactly the same as a portion of cytoplasm. Cytoplasmic structure, such as ER here, suggesting a non-selective sequestration of cytoplasmic component. We also observed the membrane sac just end up in a portion of the cytosol next to the vacuole. After completion of expansion and sealing, we could see the double membrane structure of yeast autophagosome. And this freeze fracture image showed a fusion event between autophagosome with vacuolar membrane. There, outer membrane of autophagosome is continuous with the vacuolar membrane. Here also you can see inner membrane structure, which we named autophagic bodies. Autophagic body is body occasionally contain mitochondria, shown several here and here and here. So that means autophagic autophagy can degrade not only cytosolic protein, but also supramolecular structure like ribosome, and even entire organelle, which is a characteristic feature of autophagy. This slide shows the scheme of autophagic process in yeast. When cells are faced with starvation condition, small membrane sacs appear next to the vacuole and expand to enlarge a portion of cytoplasm. Then double membrane structure autophagosome targets the vacuole and fuse with vacuole membrane, releasing inner membrane structure into the vacuole. In wild-type cell, this structure immediately disrupted by vacuolar enzyme. The proteinase deficient mutant allowed us to follow the progression of autophagy as an accumulation of autophagic bodies. The membrane dynamics of this process was essentially the same as non-mammalian autophagy, though vacuole is much larger than lysosome. As you know, yeast is a very good organ to dissection for dissection of complicated biological phenomena through genetic analysis. I was inspired by beautiful work by Lee Hartwell, which unlocked the cell cycle from, through CDC mutant, and also by uncovering of a circulatory pathway using Sec mutant by Randy Sheckman. We therefore set out to obtain autophagy defective mutant to find the gene involved in autophagy. However, at that time, I had no idea how autophagy defective mutant would behave. So we decided to perform a simple morphological screening for mutant that do not accumulate autophagic body under starvation condition. Miki Tsukada, one of my first graduate students, made a great contribution during this screening process by assessing thousands of mutagenized cells one by one for mutant lacking autophagic bodies. She identified the first autophagy defective mutant, which we named APG1-1, later renamed to ATG1. This mutant, this mutant cannot proceed protein degradation, but they grew normally in rich medium and otherwise had no obvious phenotype. However, soon we found that ATG1 mutant dies sooner than wild-type cell during long period of starvation shown here. Assuming that this phenotype is caused by the defect in autophagy, Miki ran another screen assessing the viability of mutagenized cell first and successfully obtained about 100 autophagy defect mutant. Genetics analysis of these mutant revealed 14 ATG mutant. We know now 18 ATG genes are essential for starvation-induced autophagy in yeast. So I can say this initial screen was very effective. Soon our work by electron microscopy suggested that all these ATG genes are defective in the formation of autophagosome, the most crucial event in autophagy. The next step was to determine protein encoded by these ATG genes. The first gene, ATG1, turned out to encode protein kinase, but most of the ATG genes cloned by our group and the others were completely novel. And amino acid sequence of did not tell us anything about their function. That was a difficult time for us. My lab in College of Arts and Science was quite small and had no fancy equipment. Funding was limited, and I had only a few colleagues, but it without doubt that 8 years in that lab was the foundation of my work in autophagy. In 1996, I became a professor at the National Institute of Basic Biology in Okazaki, which provided us with a very conducive research environment. I asked Tamotsu Yoshimori to join our lab as an associate professor to start work on autophagy in mammals, mammalian cells, and also Takeshi Noda and Yoshi Kamada to join us as assistant professor. The following year, Nobel Mizushima came into my lab as a postdoc. Year by year, talented postdoctoral researchers and graduate students gathered to my lab. The cloning of ATG genes proceeded much faster than I expected, thanks to the collaboration of Mariko Osumi's lab and also completion of East Gen 4 genome project. The first breakthrough was done by Nobel Mizushima. When he examined the products of ATG12, he found two bands, one of which lands much high molecular weight. He also realized he also found in ATG 5, 7, and 10, this high molecular weight band is missing. I won't go further in detail. We found that ATG12 is a novel ubiquitin-like molecule, and it's activated by E1 enzyme, ATG7, then transferred to E2 enzyme, ATG10, and finally form a bond with ATG5. And finally, this ATG12-5 conjugate is bound by ATG16 dimers. The discovery of this enzyme system uncovered the function of 5 ATG genes all at once. Another molecule we are interested in was ATG8 because immunoelectron microscopy showed that ATG8 is localized to the autophagy-related membrane as shown here. Suggesting that a good marker for membrane biogenesis. Two talented graduate students, Takayoshi Kirisako and Yoshinobu Ichimura, unveiled another unique conjugation system. ATG8 is synthesized as a precursor that is processed by ATG4 proteinase, and it's activated by the same E1 enzyme, ATG7, and then transferred to ATG3. But rather forming with a conjugate with protein, ATG8 forms a bond with one of the major membrane phospholipid, phosphatidyletanolamine. Thus, to our surprise, we found about half of ATG genes are involved into these conjugation systems. In 1998, Takeshi Noda found that rapamycin induces the autophagy, even cells are grown in a rich medium. As rapamycin is the inhibitor of Tau kinase, we therefore propose Tau kinase is the most upstream regulator of autophagy. Meantime, Yoshi Kamada and Tomoko Funakoshi studied the early step of autophagy and showed ATG13 and 17 played an important role in autophagy induction and are necessary for ATG1 kinase activation. Akio Kihara also found the autophagy requires specific PI3 kinase complex 1, which is distinct from the predominant complex 2. Within a decade of our research in NMEB, we found 2 more ATG proteins that interact with each other, as well as ATG17, that regulate ATG1 kinase activity. We concluded that total 18 ATG proteins are essential for autophagy, and those proteins can be classified into 6 functional groups shown here: ATG1 kinase complex and regulator, ATG PI3 kinase complex, and 2 conjugation system, and ATG9 sole membrane protein among ATG gene, and ATG2, ATG18 complex. Also, during our time at NIBB, Nobel Mizushima and Tamotsu Yoshimori began to study mammalian autophagy and successfully showed that ATG12 system well conserved in mammalian cell. And Tamotsu definitively proved that LC3 ATG8 homologue can be used as a very effective marker of autophagy progression in mammalian cell. At around this time, 2 of my colleagues, Hideki Hanaoka and Koki Yoshimoto, succeeded to identify ATG genes in plant cell. Although the homology of ATG genes are low, but ATG system is well conserved from yeast to mammals and plant, indicating that the core machinery of autophagy are acquired at an early stage of evolution. The identification of ATG genes essential for autophagy changed the entire landscape of autophagy research. One aspect of this evolution is that we can now visualize the process of autophagy by fluorescence microscopy using ATG protein as markers. Nobel Mizushima took advantage of this when he constructed a transgenic mouse expressing GFP-LC3. This tool was allowed to us to assess how much autophagy occurs in every organ in mouse body. This mouse has been distributed all over the world. Nobel also developed the first ATG1 knockout mouse, and which allowed to show autophagy plays crucial roles for survival at birth. And Masaki Komatsu showed by making a conditional ATG7 knockout mouse, we showed that autophagy is essential for defective mutant accumulate ubiquitinated protein and ultimately tumor in the liver. From this point, manipulation of ATG genes in various organisms, different cell type, variety of tissue and organs, and the individuals started in many labs around the world and revealed a vast array of physiological function of autophagy. The relevance of autophagy in various diseases are getting clear. This rapid expansion of autophagy would not have been possible without a concerted effort of autophagy researchers all over the world. But this huge area falls outside the scope of my talk today. I mentioned earlier that nutrient recycling is the most primary function of autophagy, which is evolutionary adaptation to a persistent challenge of nutrient limitation, starvation. Recent work has shown that another important function of autophagy, in addition to the bulk recycling of portion of cytoplasm, autophagy is also able to pick and choose excess or harmful material from the cytoplasm in a selective manner. Target of this degradation includes specific protein, supramolecular structure, protein aggregate, organelle, and even virus and invasive bacteria. Now, it is well accepted that autophagy plays an essential role to maintenance of the cellular homeostasis. Defects in this quality control may cause various abnormality, health problems, and disease. This kind of autophagy is generally not random, but selective. Also using yeast, Dan Karolinska has studied CBT pathway, the first identified model system of selective autophagy. In yeast, several receptors for selective autophagy has been found, as shown here. Now, various selective mode of autophagy has become one of the most exciting fields in autophagy research. We are now beginning to arrive at the clear understanding for some of these selective types of autophagy, such as mitochondrial autophagy. But this is an area in which we need more research to obtain the precise molecular details. Even in the simple yeast system, many fundamental questions remain to be answered. For this reason, my group is still working on yeast autophagy. Although we have identified the core component of the autophagosome formation, the question of where and how these proteins function in the cell remained. Kuninori Suzuki made a major contribution on this area. First, he found that autophagy is induced most ATG protein localized at least partly a dot structure next to the vacuole. He also showed that autophagosome is generated from these dot structures. We call pre-autophagosome structure the path. We systematically analyzed the mutual relationship between ATG protein to uncover how the path is organized, and found the causal relationship among 6 functional groups as shown in this slide. ATG7, ATG1, 13 complexes, and then ATG9 comes, something like that. Okay. And recent improvement of the fluorescent microscopy, wow, sorry. I made a mistake. Yeah, recent microscope showed this is a ATG17. That means a dimer form of ATG17, 2931 complex. As you see here, ATG17 is just moving around in the cytosol and under growing condition. Oh, sorry. Wow. Wow. What's happened? Sorry. Wow. What should I do? Sorry. That won't work. Someone help me? Sorry. Could you help me? Okay. Okay. So, sorry. Under starvation condition, 80. Oh, I was. Sorry. Under starvation condition, a small portion of ATG 17 complex come to the pass, as shown here. That means only a portion of protein doing autophagus formation under starvation conditions. But now we know that the hierarchical relationship between ATG protein reflects the temporal sequence of events that occur during autophagus formation, shown here. Close and long-term collaboration with Nobuo Noda and the late Dr. Fuyuhiko Inagaki successfully provide us with structural information of many ATG protein and their complex, which was really crucial for our understanding of their function. Now, I'd like to talk briefly about the recent work on the early step of autophagus formation, which were mostly done by Hayashi Yamamoto and Nobuo Noda. ATG13 is a key player that which has a quite unique structure that is made in a terminal globular domain, and the rest is just long denatured, naturally disordered region. In this region, we can find many phosphorylated residue by TOC kinase. Under starvation, these ATG13 residues are quickly dephosphorylated, which allow to bound to ATG1 and ATG17 via specific site in disordered region, resulting in the formation of dimer of ATG1, 13, 17, 29, 31 complex. As you see here, we found the specific binding site of ATG13 to ATG1, and also we could identify the binding site of ATG17 to the ATG13 and in the region of disordered region. So ATG13, this phosphorylated ATG13 can bound ATG1 and ATG17, 29, 31 complex. But then Karolinska's group showed the real actual number of ATG protein at the pass is about 30 to 40. So the next question we had is how this higher ordered structure is formed. Recently, we found another binding site of ATG17 in the disordered region. And I will show our image of the early step of autophagus formation. As I said, ATG13 has a long disordered region, and after the phosphorylation of these residue allowed to bound to ATG1 kinase at terminus, and this region is a binding site of ATG17. It formed this kind of complex. And we found another binding site of ATG17. It's bound to another ATG, this complex. So that allowed to the forming a higher order of this structure. That means we can have more multimeric form of this ATG1 complex. This is important for activation of ATG1 kinase. These adjacent ATG1 by this autophosphorylation. And also we found recently, Suzuki showed the ATG N-terminal domain bound to the ATG9 to recruit ATG9 vesicle. So this is our image of the early step of autophagy, autophagosome formation. So these results show the ATG path is not a static or a stable complex, and path is flexible supramolecular assembly made up many ATG complex and also membrane structure. And path assembly is also highly regulated by modification and transient interaction of ATG protein at each stage of membrane formation. Okay. Now, at present, my group is focused on the physiological meaning of autophagy in yeast. In a way, I feel we have come full circle by going back to the original question, when and how and what is regulated by autophagy. By using latest analytical tools, Han and Tomoko Kawamata showed that massive RNA is regulated via autophagy, and the resulting base transport out from the cell. It's not a recycling system. So I believe we need more knowledge about the degradation process and also assess the degradation product of the autophagy and how they are transported out from the vacuole to the cytoplasm and how they affect the cellular metabolism. So those are questions we are trying to. So my autophagy research was always been driven by nothing more than intellectual curiosity and thirst to get better understanding of life through protein dynamics in the cell. When I started my work, I never thought autophagy becomes so relevant to disease as diverse as neurodegeneration, infectious disease, cancer, and others in such a short time. But now autophagy research has become a major field in biology. And this figure presents that the number of papers related to autophagy is so rapidly increasing. It goes without saying that this is thanks to the large part to the tremendous work, effort of many researchers all over the world. So I'd like to express my sincere thanks to share this honor with all of them. However, this figure also tells us it takes a while to establish one field. Truly, original discovery in science are often triggered by unpredictable and unforeseen small findings. Nowadays, distance between basic discovery and practical application is getting closer. While it is exciting, but scientists are now increasingly required to provide evidence of immediate and tangible application of their work. It is truly my hope that society is able to nurture not only purpose-oriented science, but also science as a core of cultural activity. If my small idea and decades of work had made a contribution to fundamental science through autophagy, and Nobel Foundation recognized the basic nature of this work with this prize, it is honestly my great pleasure and satisfaction as a basic scientist. Finally, I must acknowledge all good fortune I have had throughout my career, the excellent colleagues who have joined me on this journey, great effort of many collaborators, good friends, and continuous support of many grants, and my caring family, especially my wife, Mariko. Thank you for attention.